Bismillahirrahmanirrahim. We are here to discuss the next topic, that is host defense against the tumor, that is tumor immunity. This is about the topic. Now we are going to start it. Host defense against tumor, that is tumor immunity. Tumor antigens or flu type, that is tumor specific antigen, that is present only on tumor cells, not on normal cells. And tumor associated antigens are present on tumor cells and also on normal cells. Normal cell display multiple MHC self-associated antigen, no T cell response, while tumor cell express different types of tumor antigen. They are recognized by CD8 plus T cells. They are of 1, 2, 3, 4, 5, 6, 7 types. Product of mutated oncogene and tumor suppressor gene. Neoplastic transformation result from genetic alteration that will lead to expression of cell surface antigen that are seen as non-self by immune system. For example, HER2 neo oncogene, highly expressed in breast cancer. Oncogenic product may be mutated RS or BCR-ABL fusion product. I have already discussed this BCR-ABL fusion product in my third video of neoplasia. That is, there is a translocation, fusion gene translocation between the chromosome number 9 and 22 that lead to BCR-ABL fusion gene product, and this occur in chronic myelogenous leukemia. While tumor suppressor gene, for example, mutated p53 protein activity, we have also discussed p53 that it is known as guardian of genome. Mutated gene products, for example, various mutated proteins in carcinogen or radiation-induced animal tumors, various mutated proteins in melanoma, no self-tolerance against them. The third one is overexpressed cellular proteins. Overexpressed cellular proteins can also lead to different types of response by CD8 plus T cells. For example, tyrosinase, an enzyme induced in melanin biosynthesis. If it is overexpressed, then it will act like a tumor, or MART in melanoma. This was overexpressed. There can also be aberrantly expressed cellular proteins. The fourth one is tumor antigens produced by oncogenic virus. For example, virus produce proteins that are recognized as foreign by immune system. Most potent is DNA viruses produce proteins, for example, HPV or EBV, Epstein-Barr virus, common in most of the lymphomas. Oncofetal proteins are those proteins, for example, carcinoembryonic antigen and alpha-fetoproteins that are expressed during embryogenesis. Means they are not supposed to be re-expressed in the normal adult, they are only expressed in embryogenesis. If these oncofetal antigens are again re-expressed in normal adults, they will lead to tumor, for example, re-expression in colon and liver cancer. Then comes altered cell surface glycolipid and glycoproteins. Mucins are most important in it. For example, CA125 and CA199 expressed on ovarian carcinomas, and MUC1 expressed in breast carcinoma. What is this MUC1? It is an integral membrane protein that is expressed only on the apical surface of breast ductal epithelium, a site that is relatively sequestered from the immune system. In ductal carcinoma of breast, this MUC1 is expressed in an unpolarized fashion and contains new tumor-specific carbohydrate and peptide epitopes. Last one is cell type specific differentiation antigen. For example, lymphoma may be diagnosed as B cell derived tumor by detection of surface marker, that is CD20. What happens that in this tumor express the molecules that normally are present on the cells of origin. These antigens are called as differentiation antigens because they are specific for particular lineages or differentiation stages of various cell types. For example, this CD20 is used as a marker for the detection of lymphoma. Let's discuss anti-tumor effector mechanism. Generally, cell-mediated immunity is the dominant anti-tumor mediated mechanism in vivo. Cells show different responses. For example, cytotoxic T lymphocytes are there, natural killer cells are there, macrophages are there, and humoral metastasis is there. Cytotoxic T lymphocyte, in human they play a protective role against the virus-associated associated lymphomas. For example, EBV-induced Burkitt's lymphoma, MHC-restricted CD8 plus cells can kill autologous tumor cells. Natural killer cells are capable of destroying tumor cells without proper sensitization. They are the first line of defense. And T cells and natural killer cells provide the complementary anti-tumor mechanisms. Activating receptors are also present over there, for example, NKG2D. Macrophages. Classically activated macrophages of M1 type exhibit cytotoxicity against the tumor cells. T cells, natural killer cells, and macrophages may collaborate in anti-tumor reactivity because interferon gamma activates the macrophages, is a potent activator of macrophages. These activated macrophages may kill the tumor by either the production of reactive oxygen species or the secretion of tissue necrosis factor. This reactive oxygen species production and tissue necrosis factor will ultimately lead to the anti-tumor effector mechanism. Humoral mechanisms is also there, for example, a monoclonal antibody against the CD20. A B cell surface antigen is used in the treatment of non-Hodgkin lymphoma. Let's discuss the immune surveillance and immune evasion by tumor. Immune surveillance was a term that was given by Lewis Thomas and Burnett, and they both formed this concept that refers to recognition and destruction of newly appearing tumor cells which are seen as foreign by host immune system. It is proved by the fact that 5% of persons with congenital immunodeficiencies develop cancers. Meaning immune system is there to protect a person, and if a person's immune system is not working or they are already having the congenital immunodeficiencies, they are 5% more likely to get a chance to have the cancer. That is almost 200 times rate, that is almost 200 times reported rate for persons without such immunodeficiencies. Immune evasion by tumor means that how immune system escapes such mechanisms. When cytotoxic T lymphocyte is there, natural killer cells are there, macrophages are there, humoral mechanisms are there, how immune system escape all these things and get into the trouble or develop the neoplasm. The fact is that selective outgrowth of antigen negative variants are there, a loss or reduced expression of histocompatibility complex is there. For example, cell fail to express normal level of human leukocyte antigen and they escape the attack by cytotoxic leukocytes. Such cells, however, possibility is there that they may be triggered by natural killer cells. Immune suppression is there, for example, expression of tissue growth factor beta or expression of FAS ligand. If you remember, I have told you in earlier videos that FAS ligand was there present in the evasion of apoptosis. So the cell may be entering the apoptosis, that is the immune cell may be undergoing the apoptosis. So this is the immune suppression. Antigen masking means that tumor cell produce thicker coat of glycocalyx such as salicylic acid containing mucopolysaccharides than normal, and as a result of this thick coat may block the access of immune cells to antigen presenting molecules, thereby preventing the antigen recognition and self-killing. Downregulation of costimulatory molecules means that costimulatory molecules are required to initiate the strong T cell responses. So basically what do the tumor either inhibit or decrease the co-stimulatory molecules, and as the co-stimulatory molecules are not working, so no T cell response is established. That's all from anti-tumor effector mechanisms.